My name is Esra. I'm a doctoral student at Berlin School of Mind and Brain and also Max Planck Institute for Human Cognitive and Brain Sciences in Leipzig in Germany. And I want to first thank you all for taking your time to listen to me today. And in my talk today, I will present the effect of heart-brain interactions on somatosensory perception and Evoked-Potentials. Let's start with some background information. The brain and other bodily organs, such as heart, are in constant communication with one another. The brain receives and responds to visceral signals from the body. Our brain integrates the interoceptive information from the body with exteroceptive information about the external environment provided by the sensory organs to maintain the balance and homeostasis in the body. In this process, the heart plays an important role. There is a constant information flow between the brain and the heart, and due to this dynamic interaction, it has been questioned in some studies whether cardiac activity can influence our conscious perception. Cardiac activity occurs in a cycle with two phases known as the systole and diastole. Systole occurs when the ventricles contract and eject blood into the arteries, whereas the later phase, diastole, occurs when ventricles relax and the blood returns back to the heart. Our research group previously showed that the detection of somatosensory stimulus decreases if it's presented during the systole phase of the cardiac cycle. Following this interesting finding, in the current study, we wanted to confirm decreased detection during that systole, but also we wanted to ask whether this cardiac activity influenced perceptual discrimination ability, sensitivity, or decision bias, called criterion in signal detection theory. In other words, we wanted to know whether people detected stimuli less during systole, since their decision bias changed and they became more conservative in their detection responses, and reported more often, no, I don't feel it because of this detection threshold basically, or whether their ability to discriminate the presence of stimulus from the surrounding noise decreased during systole. Second, we also wanted to test whether cardiac activity affects early or later components of somatosensory-evoked potentials, which are neural responses evoked by somatosensory stimulus. Finally, we wanted to investigate whether neural responses to heartbeat, known as heartbeat-evoked potentials, also influence somatosensory processing or not. How did we answer all of these questions? So what did we do in our experiment? In the beginning of the experiment, we placed EEG electrodes to measure brain activity and ECG electrodes to measure the heart activity. And then we put two ring electrodes to the index and the middle finger of the left hand of the participant. Afterwards, we determined a very weak stimulus intensity that participants would only feel half of the trials. Then, we started with the experimental paradigm. In the beginning of every trial, participants saw a black fixation point in the middle of the screen for 600 milliseconds. Afterwards, we presented very brief, sudden electrical stimulation, either in the index or in the middle finger. 600 milliseconds after the stimulation, we asked the subjects whether they felt the stimulus or not. We asked them, did you feel anything? And they answered as yes or no. After the yes-no detection question, we also asked subjects that in which finger they think there was a stimulation. So we asked them, even if they didn't report feeling the stimulus in the first question, we still asked them to localize the stimulus. One thing important in the experiment that I should mention is in the experiment we lied to participants and told them there will be always a stimulation in every trial. However, 20 out of 100 trials in every experimental block were null trials, which didn't contain any stimulation at all. In total, there was 8 blocks, and we tested 37 healthy subjects in the experiment. And afterwards, we checked whether perception varied across the cardiac cycle. In order to do this, first, we determined whether stimulus onsets occurred during systole, which is this early cycle indicated with red, or in diastole, this blue-colored area. So we checked where the stimulation occurred in the cardiac cycle, and we classified each stimulation as systole trial or diastole trial. In our study, we determined the systole window from the R peak, which is in here, until the end of the T wave here. And we use a specific automated algorithm to determine this for every cardiac cycle of each individual subject. And after determining the systole window, then we chose an equal length of diastole window from the later phase of the cardiac cycle. And we did this redefinition of diastole window to keep its length equal with systole window so that we could have equal likelihood of having a stimulus in both windows. After determining this, we calculated correct detection rates when stimuli occurred during systole, which is the red, which is shown as red here, and then diastole, which is blue here. We confirmed that the somatosensory detection would be lower in systole. Then, by using signal detection theory analysis in each cardiac phase, we calculated sensitivity. In other words, perceptual discriminating ability of subjects. We found that less detection during systole was indeed associated with less sensitivity of subjects to the somatosensory stimulation during systole. So there was a significant difference between these two distributions. After this, we wanted to also explore the relationship of this cardiac effect with the pulse wave, which is temporarily coupled with the onset of the systole. In an exploratory analysis, we investigated the effect of the time delay of somatosensory stimulation from the previous R peak on detection and localization rates. We found that specifically in this time window, which is between 200 to 400 milliseconds after the R peak during systole, there was a sudden dip in the correct detection and localization rates. This is the time window when the pulse would arrive to the fingers as well. This finding was quite interesting for us because it brought a clue why we would observe the cardiac cycle effects on somatosensory perception and helped us to interpret these results in a predictive coding framework. In this framework, the heartbeat is treated as a predictable event and it is largely ignored by the brain. As we found that perception will be lowest when the blood pulse will be maximum in the fingers, we concluded that weak external stimuli at the finger may be mistaken as heartbeat pulse-associated noise and ignored by the brain. We tested this argumentation by looking at the associated neural activity. So we looked at the EEG data afterwards to investigate neural correlates of this cardiac cycle effect on perception. In our analysis, we focused on event-related potentials following stimulation at C4 electrode, which represents contralateral somatosensory region here. We tested whether there is a significant difference in neural responses to stimulation during systole and diastole phase of the cardiac cycle. So here, the blue line is showing the trials which happened during diastole, and the red line is showing the somatosensory-evoked potential for the trials happened in systole. And we wanted to understand whether the brain responses to stimulation differed depending on stimulus timing during the cardiac cycle here. And in order to test this, we ran cluster statistics for this whole time window, 600 milliseconds time window after the stimulation. And what we found was that we found two significant clusters with a significant difference between systole and diastole conditions, which was starting from 276 milliseconds to going up to 468 milliseconds here. This difference occurs in the time window of P300. which is a specific component of brain activity associated often with consciousness and also predictive mechanisms in the brain. And after doing this analysis in the sensory level, we wanted to ensure that this P300 evoked potential difference we observed in the sensory level was coming from indeed primary somatosensory areas. In order to do that, we ran source reconstruction analysis. Here, on the right side of the plot, you see the source level analysis, basically, the results. And we selected a region of the somatosensory cortex, which is representing the finger, left fingers, basically. And then we looked at the trials which happened during diastole and during systole, and we looked at the source level somatosensory-evoked potentials here. And we confirmed in source level as well that there was a significant difference in late components of somatosensory neural responses in the contralateral primary somatosensory areas. So, decreased P300 responses during systole, in addition to less sensitivity during systole, strengthened our argument that predictive mechanisms in the brain might be responsible for cardiac cycle effects. And additionally, I want to add that we controlled for the cardiac pulse artifacts in the data, and all the results that I showed so far were cleaned from such kind of artifacts. Because one can assume that just due to movement of electrodes by the blood movement, blood fluctuation in the vessels might create some differences in the neural activity. And for example, if one, let's look at the ECG activity here, and 0 corresponds to basically stimulus, somatosensory stimulus onset here for systole and diastole. And just because of the movement of the blood itself, there will be a significant difference between these two conditions, basically. And we run a specific algorithm to estimate this kind of pulse artifact in the data, which is shown in the middle panel here. This one, E, is showing it for the heart electrode, and B is showing for a brain electrode, which is T4 that we used. And by estimating this and subtracting this from the original raw data, we end up with, for the brain, for C4 electrode, this, which is the same plot that I showed you in the previous slide. And for heart activity, basically we canceled out all the differences between systole and diastole. So whatever, of course, the difference here became smaller, but it was still significant in the data. So even when we accounted for such kind of pulse artifact in the data, we still found a significant difference for trials which happened in systole and diastole in the brain data, basically. After answering and looking at the cardiac cycle effects on perception, we next asked whether how our brain responds to the heartbeat has an effect on our somatosensory perception. To do this, we use heartbeat-evoked potentials. So what are heartbeat-evoked potentials? Heartbeat-evoked potentials are representing the cortical processing of the heartbeat. It's a very similar concept to sensory-evoked potentials, as how we epoch the data, EEG data, according to stimulus onset. When we are analyzing sensory processes, one can do the same thing for the heartbeat. So one can take the brain data, EEG or MEG data, and then epoch the data according to the heartbeat onset, and then see the following brain responses to heartbeat. Heartbeat-evoked potentials are generally observed between 250 to 400 milliseconds after the R peak in frontline central regions. And it has been reported that when people are interoceptively attending, such as when they're, for example, doing a heartbeat counting task, there was an increase in heartbeat-evoked potentials. So there is a positive correlation between heartbeat-evoked potentials and interoceptive attention, shown from the literature, basically. So how we did our analysis? In order to run the HEP analysis, we had to do some trial selection in our data to ensure that the time window of HEP, which would correspond to 250 to 400 milliseconds, was free of any stimulation-related activity. We only included trials where stimulus occurred at least 400 milliseconds after the preceding R peak, so which was corresponding milliseconds after the heartbeat, or 200. That the heartbeat evoked potential would be always a prestimulus activity and wouldn't be contaminated by stimulation-related activity in the brain. After doing this careful selection of trials, we looked at prestimulus heartbeat evoked potentials before HEPs. The trial subjects detected and reported, yes, I perceived, versus misses, the trials where participants said no, even though there was a stimulation, basically. So what you see here is that we look at the heartbeat evoked potentials in these brain regions. We found that in these brain regions, following 296 to 400 milliseconds after the R peak, there was a significant difference in heartbeat evoked potential between preceding misses and HEPs. So here the dark line here is showing the heartbeat evoked potential preceding misses, and the lighter green line here is showing heartbeat evoked potentials preceding HEPs. So as you can see here, that preceding misses, the heartbeat evoked potentials were higher. After doing this analysis, also we wanted to understand whether this detection difference is connected to a sensitivity or decision bias according to signal detection theory. In order to run this analysis, we used binning approach. So what we did was we calculated an average heartbeat evoked potential for preceding every trial for this time window and for this cluster electrodes here, and we calculate one average value, and we sorted them from low to high. This is another way of demonstrating what I showed in the previous slide. So as the heartbeat evoked potential increased, the detection rate decreased significantly. And afterwards, using signal detection theory analysis, we found that this effect was connected with increasing criterion here. So increasing heartbeat evoked potentials was followed an increase in the response bias, meaning that people became more conservative in their decision biases, and they reported more often, no, I didn't feel the stimulus, therefore. A conservative bias is known to associate with decreased baseline firing rate across different brain regions. Supporting this mechanism of criterion, we found that following high HEP amplitudes, which is shown with the dark blue here, there was a decrease in somatosensory-Evoked potentials starting from the early components. So starting from the P50 activity and later components here, there was a significant decrease in the somatosensory-Evoked potentials following higher levels of heartbeat-Evoked potential. And I want to add here that we also observed this decrease in early component of P50 in right anterior insula, posterior cingulate cortex, lateral prefrontal cortex, the areas known to modulate external versus internal attention. Given that HEP amplitude has been shown to be significantly higher during interoceptive compared to exteroceptive attention in previous studies, we propose that the modulation of HEP amplitude reflect attentional shifts between external stimuli and internal bodily states. In other words, a large heartbeat-evoked potential seems to reflect a state of mind in which we are more focused on the functioning of our inner organs, such as blood circulation. However, we are less aware of stimuli from the outside world. And also, just to add up that we also check in this data and in this analysis, we control for cardiac field artifact, and we ensured that this difference in heartbeat-evoked potentials were not caused by a difference in the electrical activity of heart itself preceding misses and hits. Here you see the ECG activity preceding hits and misses, and there was no significant difference in the heart activity itself, but it was we specifically observed it in the brain electrodes, which means that our HEP effect that we observed is not attributable to the changes in the electrical field of the heart itself. And after doing all of this analysis, we also finally wanted to check another rhythm, which is called alpha rhythm, which is known to influence sensory processing. And we wanted to see whether alpha rhythm has an association with these heartbeat-related effects. Just to give a small recap, that in the previous literature, it has been shown that as alpha amplitudes are going higher, the detection rates and localization rates known to go lower. So higher alpha is associated with lower detection rates. And we wanted to know whether given the similar type of alpha power, whether the heartbeat-related effects would still have an effect on detection. So therefore, we did again binning analysis, and we sorted alpha power from the low to high amplitudes. And then we looked at the systole and diastole trials and their detection rates for similar levels of alpha. And still, given the similar levels of alpha, we found significant differences between systole and diastole trials on detection. And we also did the same analysis for HEP, heartbeat-evoked potentials. And again, for similar levels of alpha, we still observed a significant difference between low and high HEP levels on detection. And our analysis shows that neither of two heartbeat-related effects on perception was mediated by Prestimulus alpha amplitude, but rather both are independent and additive to the effect of Prestimulus sensory motor alpha amplitude. So to summarize our results, we found that participants are less sensitive to somatosensory stimuli during systole. Secondly, we found that P3 component of somatosensory-evoked potentials are suppressed during systole. And finally, increased heartbeat-evoked potentials were before stimulation is followed by decreased somatosensory detection, as well as decreases in early as well as late SEP amplitudes. And as next steps, we want to also do some further analysis, and we want to check functional connectivity and fMRI activations across the cardiac cycle. Also, we are thinking of looking at alterations of cardiac phase dependence in subjects with hypertension. And currently, we have a project where we are looking at heartbeat-evoked potentials in cardiac disorders such as atrial fibrillation. I want to end my talk by thanking to my collaborators, and specifically to my supervisors, Vadim Nikulin and Arno Flieringer. And Arno wanted to also join today, but he had to join to another meeting, but he asked me to send you all his regards, so I'm doing that. And thank you all for listening to me.